This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 115, recorded January 6th, 2011. Hey everybody, it's Vincent Racaniello, and it's time to talk about viruses. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
How are you, Alan?
Doing okay.
You know it's going to snow again.
Yeah, we're supposed to get some little inch or two, something like that.
All right, not major snow like last week.
No. Well, that wasn't even up here. That wasn't really major snow. It was only 8 to 10 inches.
Also joining us today from North Central Florida is Rich Condit.
Hello there, fellas. How you doing?
We're okay. How are you?
I'm good.
Is North Central Florida, in Gainesville in particular, good today?
Yeah, it's good. We got clear blue skies, 63 degrees.
All right. Well, hoping you can stay with us for an hour and not go tee off, okay?
Yeah. I can hang out. Yeah,
all right. Our special guest today from the wonderful podcast Futures in Biotech, which is a TWiV podcast, Marc Pelletier. Hey, Marc.
Hello. Hey, this is amazing. You've assembled a group of scientists with radio voices.
You think?
It's awesome.
I don't know about me, but those guys, Rich and Alan, are amazing.
Yeah, absolutely.
You do okay, Vincent. You're right in there.
Yeah, you sound good.
Marc is the creator of, one of the oldest podcasts I know. I actually been called out about that. It's not the oldest, but it's quite old. When did you start Futures in Biotech?
In 2006, of June 2006. It was one of the first of the TWiV podcasts.
I see, yes.
Back when it used to be a blog, and Leo did the editing.
He did the editing, really? Wow. Well, if you haven't checked out Futures in Biotech, you should at twit.tv/fib. He always has great scientists. You have a different style from TWiV. You get different hosts, different guests every week, different scientists, and you also cover all of biology, really.
We try to. It's a different format. Yeah. It's easier to prepare for an interview when it's the guest that does all the talking, right? So that's what kind of my reason.
That's always my strategy.
What you have to do is be curious. You listen. Listening is the first thing, right? If you don't listen, you can't ask good questions.
Exactly. It also, what a great way to network. I mean, you get the opportunity. Most people don't say no, and there's some great scientists out there that there's very little place in the sort of blogosphere or the in the libraries of audiovisual that has an audible record of what's really going on in their mind. If you watch Nova or PBS, it's usually sound bites that TV usually produces.
Yeah.
So I try to give people a first-hand account of some of the great science that's going on.
No, you do really good, well, because you get deep down into it, you drill down, and you really explore the science. I really enjoy it.
Sometimes I miss it completely.
No, not usually. I liked Bonnie Bassler. She was one of the recent ones.
Yeah, that was struggle.
You had sound problems there.
Oh my God! She's so popular at her school that the students were using up the bandwidth, and we could hardly get a connection. Mm-hmm. And I was just cringing the whole time. It was really tough. But we'll get her back.
The cool thing about Fib is that you do live video at the time of recording, and then you record it and you put it out both as video and audio, and so people can watch it. So today, one of the papers we're going to do today is from the lab of Lynn Enquist, and he's a colleague of Bassler's at Princeton, of course. And I told him, hey, we're going to do your paper today. And he goes, oh, when can I watch? He probably figured it was a fib. But it's not. So it would be nice someday to do video. I think it would be fun. It brings another element, right?
I think you could do it with Ustream and create and if you do it, I would do it on a Friday, somewhere between 2:30 to 3:30, or from 4:30 to 5:30, mm-hmm. Just around Fib, or before, so that the listeners could just tune in and sit down and listen to 2 podcasts.
Oh, you don't want us to conflict with Fib, do you?
No.
Well, we usually start at 2 p.m. and we're done by 3:30, and that's when you start. So that's pretty good.
Yeah, it'd be awesome.
But you and I try and go back and forth on our shows, and I think it's a good way to have our audiences learn about the other podcasts.
You're a resident subject matter expert with respect to virology.
I love being on Fib. In fact, today we have an email which refers to my last appearance on Fib, so we'll get to that later. But we have 2 cool stories today. The first one I had already in mind last week, and it's a really cool gee whiz story. The second one is a little bit of engineering, which I think you'll like, Mark. So let's do those. First one is a paper from the lab of Lynn Enquist, and let me get it up here. It is called Herpes Viruses Carrying a Brainbow Cassette Reveal Replication and Expression of Limited Numbers of Incoming Genomes. And the authors are Kobler, Lipman, Thurkelson, Dobashes, and Lynn Enquist. Now, the question here is something I've always wondered about, and maybe, Rich, you did too. So you take a plate of cells, and you put some virus on them, and you let it replicate, and then you measure the virus. If it's polio, you get 10 to the 9th plaque-forming units per mil in the medium after so many hours. If you put 10 times more virus on to start the infection goes faster, but you don't get any more virus. If you put 100 times more virus on to start, the infection goes even faster, but you still only end up with 10 to the 9th per mil. Now, is that the same with pox, Rich?
Yeah, pretty much. As a matter of fact, I've always worried, I don't know that I can document this, but I've always worried that if you actually had too much virus, you hurt the cells so bad that your yield might even go down.
That's right.
For normally growing virus, I usually choose a low multiplicity of infection, not only because of this issue of potentially hurting the cells, but also because of issues with the von Magnus effect, which we could talk about. It winds up not applying to vaccinia, but generally passaging virus at high multiplicity can be a bad idea. Right, at any rate, yes, I've wondered about this at the same time. I've always figured that cells just had a limited capacity to put out virus, but this paper raises other issues that are very interesting.
Yeah, and it's always kind of in the back of your mind with something like that. Well, why does it work that way?
Mm-hmm. And actually, for listeners who aren't familiar with this, the whole multiplicity issue does become an issue. As an experimentalist, you get to, in the laboratory, set up your infections where you have a certain amount of virus, you got a certain number of cells on a dish, and you get to design the experiment as to how much virus you want to add to those cells. And we call that multiplicity of infection. How many virus particles per cell? And depending on what you want to do, you may want to use a very low multiplicity infection, where in fact you have fewer virus particles than there are cells, like 0.1 or something like that. Or you may want to use a higher multiplicity of infection. And in particular, if you want to do some sort of biochemical experiment, you want a fairly synchronous infection. And under those circumstances, you want to infect every cell. And as it turns out, it takes a multiplicity of at least 3 viruses per cell to get 95% of the cells infected.
So this paper, I think, didn't start out as addressing this question. I think when they began this work, it was probably accidental, but they end up answering this question of why is it that you can only get so much virus out of a cell?
Yeah, I wonder what their ultimate motivation is. I got a hunch they're going to stick this into mice, mice eventually. Okay,
no, exactly. I think in fact, I think they've done that. And then they got some interesting results. So the virus they use here is called pseudorabies virus. Now, this is a herpes virus, even though it's got rabies in the name. It has nothing to do with rabies virus. It's a swine herpes virus, and it actually infects a lot of mammals in addition to swine. But the Enquist lab uses it because it moves along neural pathways, and you can actually trace neuronal circuits with this virus. So you can put a fluorescent protein marker in the virus and then infect animals or inject the virus into specific places, and you can trace neuronal circuits. It's really beautiful.
Did we talk about this on an earlier TWiV?
I don't think we
so.
have. No, I don't think we have. And
I knew I'd seen it before or talked about it before or something, but it didn't.
We may have mentioned it, but I don't think we've ever, done like a real piece on it.
So this is actually a good reason to have Lynn Enquist back on TWiV to talk about neuronal tracing with this virus.
Right. So importantly, herpes viruses are double-stranded DNA-containing viruses, replicate in the nucleus. They make an icosahedral capsid. It's also enveloped, and they are pros at establishing latency so that they can stick their genomes in the nucleus of cells and not integrated with the chromosomal DNA, but as a separate episomal piece, and lay there in a quiescent state for a long period of time, and under appropriate conditions become reactivated and make more virus. And some of these viruses are transmitted around the organism through neurons, as is pseudorabies. And as you've already said, Vincent, they make handy tracers in that regard.
Now, the technology, we have to take a little bit aside here and talk about the technology, which is called Brainbow. Instead of rainbow, you just put a B there, Brainbow. And this comes from another group who made transgenic mice with a gene, with a piece of DNA, and they call these Brainbow mice, that can make one of 3 different fluorescent proteins, depending on how the DNA is recombined. So they have a red, a yellow, and a cyan fluorescent protein gene all on one plasmid with a promoter at one end. And then, if you remember in a previous TWiV, we talked about the Cre-Lox recombinase system, where you can flank genes with LOX sequences, and they will be recognized by the Cre recombinase so that they will be cut out. And they've designed this cassette of 3 fluorescent proteins genes so that when it's put into a cell expressing Cre, there are different specificities of the LOX sequences surrounding the fluorescent protein genes. And you randomly get 1 or 2 or a third color produced, either 1, 2, or 3, either red or yellow or cyan. So the recombination occurs once the DNA is in the cell, and then that's it. It only happens once. And then you get, and there's a figure that you gave us, Rich, from that accompanying paper, or the paper that described the Brainbow system, where they show that in a cell culture where you've put in this plasmid, you get cells of 3 different colors.
It's really cool.
This is one of the prettiest.
And the cells will inherit the color of their mother cell.
Right. Yes. Once it's blue or yellow or red, that's it. Right. And
so all descendants of that cell will then be marked in that color.
So if they made mice with this cassette.
Actually, if you're red, it seems to me you still have the potential to become yellow or blue, right?
Yes, you're right, because you have all the LOX sites, yeah.
So if you're red, you are multipotent in that regard, okay? But once you are yellow or blue, you're stuck.
That's correct.
Okay.
So that's the Brainbow mice, and they used it to differentiate neurons, actually. So adjacent neurons would be different colors.
Hence the name Brainbow.
Yeah. So it's cool, right?
Yeah. Very cool.
And so then the Enquist group decided, let's take this Brainbow cassette and put it into pseudorabies virus, and we can then put it in mice and trace neurons of different colors. So what they say here, preliminary circuit tracing studies using this Brainbow virus, which they call PRV263, revealed a surprising number of single-colored neurons that were in synaptic contact with Cre-expressing neurons. This result suggested that only a small number of viral genomes were spreading from Cre-expressing neurons to presynaptic neurons.
Yeah, I think what they expected was that you would get the recombination in an infected cell, so you'd have 3 different colors of viruses, right? Mm-hmm. And then all of those would be transmitted to adjacent cells.
Right.
That's what they expected. And instead, what they saw was you got cells that were unique in color, as if you weren't getting a lot of virus transmitted.
Right. So it actually seems like this paper sort of stemmed from initially an incidental finding.
Mm-hmm.
Yeah, they found this in mice. They wanted to use this virus to trace circuits, right? And they found, it's not as many colors as we think. So let's follow this up. Marc, you okay so far?
Yeah. It's pretty amazing stuff.
Well, you like transgenic animals, right?
Absolutely.
You work on one in your company.
Yeah, we're always looking ways to knock down a gene in the brain.
So this may be of use if you ever want to color cells differently. You might think about using these rainbow cassettes.
Yeah, it's pretty wild. We need to go into larger mammals. The non-genetically tractable animals. Mice are pretty easy to do.
How big? Like pigs? You can make transgenic pigs.
Yeah, we're working on head trauma, so we want an animal with a soft head. large animal, soft head.
I got some friends I could give you.
It tastes good after. Yeah.
What animal has a soft head?
Well, not pigs, right? Pigs.
Yeah, they have a hard head. Yeah.
Yeah. You just want to work with a non-bunting animal, and goats, for example, they're bunting animal. All the animals that you'd think. I suppose kitty cats. I don't want to. Let's maybe for another discussion.
Okay. Anyway, this could be useful from anyone who's doing transgenic work and wants to track cells. So they have they made this virus which has this cassette, and when you infect a cell with this virus, you can get any of the three color viruses made. So they have this very cool growth curve in Figure 1.
Yeah. Where
at each time point after infection, they look at the color of the virus that's produced. And very early on, you just have red virus because that's what you put in. So without any recombination, the virus is red. That's correct, Rich?
Yep.
Right.
And then as the virus replicates, at each time point, they look at what colors, and all three colors, the yellow, the blue, and the red are present. So
and you get about equal quantities of the yellow and blue, which shows that event is not discriminatory.
Right. Mm-hmm. So of course, at each time point, you have to assay the virus not only for the titer, you have to look at the color.
Right.
Which of course you can do under a fluorescent microscope. Right. You just say, what color is this virus? So this is cool. All right. So then they did it. This is the part now where it gets a little maybe confusing because
I had to read all of this stuff about 5 times because it does get confusing.
Yeah, it's pretty dense.
So they do an infectious center assay. All right. So you infect cells with this rainbow virus, and then before the cells break up and release virus, you take those cells and plate them on a second monolayer. And that second monolayer doesn't have Cre in it, so no more recombination is going to occur.
Right.
So wherever a cell sits, it will release virus and make a plaque. All right, so plaques are started by the virus released from an infected cell. And apparently each cell can release as many as 1,000 infectious new particles. All right. So they do this experiment and they say, what do we get? Do we get all possible combinations of color in this infectious center assay? Or do we just get 3? And what they found is that one in each infectious center plaque, so that's a plaque that's getting virus from an infected cell, 1 or more cells express a single pure color, either the blue or the red or the yellow, which is surprising. You wouldn't expect with so many thousands of viruses coming out of that infected cell that you would get cells expressing only a single color.
Well, now, wait a minute. Let me get this straight. If we look at table 1, for example, at a low multiplicity, they infect the cells, they make, they take the infected cells and plate them out.
Right.
And they're seeing that like over about 70% of those guys throw off single-colored plaques.
That's right.
Okay? Right. Yellow only or cyan only. And that implies to me that at the low multiplicity, and this is actually, let's anticipate some of this, because what they say eventually, based on a lot of this stuff, is that the Cre recombination that happens before the virus has a chance to replicate its genome.
Right.
Okay? So if you have, at a low multiplicity, most of the cells that get infected are going to be infected with a single virus. And if the Cree recombination then happens on that virus, you're going to have a single genotype with respect to color, either yellow or cyan in that virus. And so if you then plate that out as an infectious center, that's going to populate as a pure population. A few of these guys, even at a low multiplicity, are going to start out either red because they haven't done any recombination, or have, say, two colors in them. But they're in a minority at that low multiplicity. 13.8% two-color plaques, 1.1% three-color plaques, and about 15% red-only plaques. So that's okay. The high multiplicity is what gets confusing. Okay? So
Right.
at a high multiplicity, the infectious center yields a lot of 2- and 3-color plaques, which I guess you would expect. That suggests that the cells initially took up more than one virus particle, which you would expect at a high multiplicity, and those two can recombine with the Cree system independently and throw off either cyan or yellow. And a much smaller percentage are red-only plaques, okay?
Mm-hmm.
And it's what grows out of those that's the big surprise to me, okay? Because this is where these movies come in. Mark, have you got these movies?
No, I don't. I do not.
They're
have
very, very cool.
Have you got the paper in front of you?
Yeah, I do. I'm looking
Okay.
at table one.
Supplementary, table one, if you pan in the paper somewhere and get supplementary figures, supplementary movies 1 and 2. So they put these infectious centers on a monolayer and do video microscopy on those, all right? And they've got these color movies of the plaques growing out from the infectious center. So I'm looking at supplementary movie 1. You start off with red cells, a couple of red cells there. So those are guys that in the infected cell, they haven't done their Cree recombination. And then as the infectious center grows out, you get cells lighting up as a plaque forms in this, more and more cells growing out from this plaque. But they are the resolved colors. Some of them are probably mixed, but you got some pure blue ones and some pure cyan ones. And the biggest surprise to them is that those happen in a sectored fashion. There are actually spots of areas in this plaque that are all blue, or areas of the plaque that are all yellow, okay? As if once you get the Cree recombination happening, there's only a limited amount of virus that's passed off in one direction. You don't get a cell with a couple of viruses. You might what they originally expected was that all of the cells would represent all of the colors.
Right.
Okay?
So they
Right.
would say that, yeah, you'd get so when you mix 3 different colors together, you get some non-pure color, basically. So the pure colors are the red, yellow, and blue. And
Right.
when you mix them together, you get non-pure colors. They thought that every cell should have a non-pure color. They would be all the
Especially
same.
at a high multiplicity.
Right. They figured you infected a high MOI, then all the viruses are expressed in all cells, and therefore you get some kind of non-pure color, but it's the same in all the cells.
Right.
But that's not what they saw. They found that each cell, as you saw in this movie, has a unique, mostly non-pure color.
Right.
And sometimes you get cells with pure colors, the original red or blue or yellow. So that suggested that something was limiting the number of viruses that could grow in each cell. Is that reasonable conclusion? You okay with that?
Right. And because the color remains constant, it also tells you that the ratio of expressing levels of the different viruses is remaining constant. So
Right.
if you have a red virus and a blue virus, or a red virus and a cyan virus in the same cell, and they're both expressing at a certain level, and it gives you a particular color,
Right.
the cell doesn't shift
It does not.
in
That's
one
right.
direction or another. And so the 2 viruses appear to express the same relative level all the way through the
Yeah.
infection.
It doesn't change through the infection. You get the same
Right.
color from start to finish. That's
Right, which allows you to look at the color and analyze, which parts of the spectrum would you put together in what quantities to get that color, and that allows you to determine the number of viruses or the number of different possibilities that you have in the cell.
Right.
I think one of the reasons that this is so difficult to think about is that once you play the movies, all you want to do is sit there and watch the movies.
That certainly took up about 15 minutes of my time when I was trying to read this paper, and then I went to the supplementary movies, and oh, wow.
It's amazing.
Mark, do you have access to the supplementary?
I don't, but I have the color pictures here, and I certainly recommend that people that are listening download the paper and look at it while during this conversation.
It's an open access paper.
Yeah,
it's open access, so you can go get those movies, Mark.
They're QuickTime movies. You can download them and just have a look. They're amazing.
Mark, if you open up the Google Doc, there's a link to the paper, and you'll find the movies. The Google Doc. But before we go on, so we'll summarize here. You have a virus of one of three colors. When you infect cells at a high multiplicity, you would expect every cell to express all of them and be the same mixed color, but it's not the case.
Right. As the plaque grew, you would expect all of them to be the same mixed color, but it's not the case.
So the rest of the paper is involved with looking at that and figure out what's going on. And as Alan said, one of the key properties was that this color doesn't change from the start to the end of the infection. Once a color is established, whether it's pure or non-pure, it stays that way.
Actually, Mark's right. In these ought to be like art gallery type pictures. Look at this, like in Figure 2.
Oh, they're like stained glass windows. They're gorgeous.
Yeah, they're just beautiful. And actually, that Figure 2A in particular, I think, is a really good example of the sectoring in the plaques. Because in the upper right-hand corner, you got something that's very green. Down in the left is something that's very blue, and you have some others that are multicolored. But you get some sectors that breed really true, indicating that they arose from a infection with maybe just one virus or something like that, and then it's propagated that way.
So then it's propagated, and that's what gives you the sector. So the green cells are all from a single propagated infection, right?
Right, yeah.
Now, the next experiment they do is very cool. They purify each color virus. So now you can plaque purify and purify a yellow, a red, and a blue virus. And then you can mix them together, equal amounts, and infect cells. And then they use flow cytometry to purify red cells and yellow cells. And then they incubate those cells and take the virus that comes out of them and ask what color is it. And to do that, they have to infect new cells. So you infect cells with a mixture of the two viruses. I think they use red or yellow. And then 6 hours later, you sort them by flow cytometry. You take the yellow cells and red cells. You take the virus that are produced by those, and then you plaque them and see what color they are.
Right.
So when you sort red cells, you get red virus out of them. When you sort yellow cells, you get yellow viruses out. So the conclusion is that the genome that's expressed in the cell is the one that's packaged, not a different color.
Right.
I suspect that people may be confused by that, but there are presumably other colors, other genomes in those cells. So in a red cell, there may be yellow genomes, but they're not being expressed. Therefore,
right, otherwise they would show up in the subsequent plaque assay.
Right. So they're not being packaged. So the way the experiment's done, it's done at a high MY, 10 or 100. So you're going to get co-infection of cells with both red and yellow viruses. But you only see cells that are red that you sort only produce red virus, even though there's probably yellow virus in them. Am I getting that right, Rich?
I think so.
Okay.
Yeah, that's my understanding of it.
So that's an interesting observation. All right. Now the next experiment is really a big one. And this is the one where the main conclusion of the paper comes from.
Right.
So they take a mixture of all three viruses, equal amounts of all three viruses, red, cyan, or yellow. And they infect cells at different multiplicities. And then they take individual cells and they determine what color they are. They looked at per condition, per MOI, they did 6,000 cells, and they look at them under a microscope with different filters so they can figure out what color they are. So they say this cell is red or yellow or blue or something in between. And they can actually go in between quite a bit with their different filters. So they plot this in a very interesting way. This is called a barometric plot, which I had not encountered before.
No, that was a new one to me.
But it's basically a triangle where at each vertex of the triangle is the pure color, the yellow or the red or the cyan. And then in the mixtures, in the middle, are all the unpure colors. And then on the vertices are the two color mixtures.
It actually reminded me of the sort of triangle inside a circle color wheel that you get in some image editing programs.
Similar. So pure colors on the vertices. Now the points of the vertices, right?
Right.
The lines are the two colors, and then in the middle are all the unpure colors.
Right. So this lets you plot. It lets you quantify a color.
Right. And they can do that by multiplicity. So they have a whole bunch of multiplicities here.
And they had it set up with a bunch of different filters that could distinguish colors that represent combinations of the other two colors.
Exactly.
So they're using a bunch of different filters here that allow them to measure shades of these combinations of these different colors, which wind up in different parts of this triangle plot.
Right. So,
by the way, the triangle plot, if you look at the actual border of the triangle, is actually data.
Yes. There are dots on
I hadn't realized that.
Yeah, those are the two color mixtures, right?
You got a lot of pure data, a lot of pure YFP, RFP, and CFP.
Yep.
It's kind of funky. It's
very cool. So here's the story. And what they say is, as you increase the multiplicity, you should get more genomes in each cell, and you should get more mixed colors. That's what you would expect. But what they found, as the MOI goes up, you get more double color mixtures, more cells on the edges, single and dual colored cells.
Well, I think actually, if you look at the data, you do, as the multiplicity goes up, you get more clustering of these things towards the middle of the triangle.
Right.
So I think you do get an increase in multicolored cells, as you would expect. But there's still a lot of guys out at the vertices. Okay, so even at a very high multiplicity, you got pure cells. So it didn't become nearly as homogeneous as they would have expected. They would have expected all of these cells to have just a mixture of all three colors, but instead there's still a lot of cells that are pretty pure.
Yeah, and they were amazed that they even saw single color cells.
Mm-hmm.
They say the probability of at an MOI of 100 of getting a single color cell is 3 × 10 to the minus 29th.
If there were no selection for, if the viruses could all replicate in that cell at the same time.
Right. But they don't.
So clearly they can't.
So this tells them that there's not every genome that gets into a cell doesn't replicate.
Right. It says that there's some kind of limit on the number of genomes that you can replicate in a cell at the same time.
Then they do a mathematical estimation of that number. And here, I cannot explain it.
Right. They do math.
They do math, and that's as far as we go as virologists. And I asked Lynn, and he said, yeah, it's math.
It's actually, it's not that complicated an equation. It's not calculus or anything, but I don't know what the derivation is for the formula. I don't know where it comes from.
Yeah, this seems to be a relatively simple formula, but I don't know where it came from either.
Yeah, I imagine it's a straightforward, some variation of a Poisson distribution put into something else, but yeah, they do some math.
So the math ends up telling them that fewer than 7 genomes. So there are two kinds of viruses they use: an attenuated virus and a virulent virus. Fewer than 7 attenuated and fewer than 6 virulent genomes are expressed on the average in each infected cell. So even if you put dozens of genomes in a cell, 6 or 7 are all that ever replicate.
Or put it actually more accurately, if you add to the cells as many as 100 infectious units per cell, what replicates is actually even 7 or fewer. Is actually only 7 or fewer. Then the question becomes, did all those guys get into the cell? Where's the limitation?
Right.
Okay, did all 100 get into the cell? And if they did all 100 get to the nucleus? And if they did, what fraction of those, was it only 7 of those then that replicated? And where's the restriction?
Right. So they think it is after getting into the nucleus.
Right.
So they think that, yeah, you add 100 genomes, and the 100 genomes get in the nucleus, but only 6 or 7 will actually replicate.
Right.
But we don't know why. I mean, that's the next step, is to figure out why.
They have some interesting speculation about that it's
very interesting.
Maybe because these the herpes virus genomes cluster in these nuclear domain 10, these ND10 bodies. And there's a limited number of those in the nuclei, and there may be some additional viral mechanism at work there too that has evolved to do exactly this.
So this is interesting, but the other interesting question is, so here we have a quantification of the observation that we first said at the beginning, that when you put more virus in, you don't get more virus out. And that is, there's apparently for pseudorabies virus some mechanism that limits the number of genomes. Right. So how many different viruses is this working in? If you did the same experiment, made triple color viruses for pox virus, polio, a retrovirus, would you get the same result, or would it be different?
It's going to be hard to do for polio.
Impossible, right?
Yeah.
You can't put 3 different colors in, unfortunately. You can just
Is that because it's too small?
Yeah, you can only put 1 fluorescent protein gene in polio. If you try more, you kill the infectivity. Yeah. And I suspect that it's a function of, the genome is small, so you can't go above a certain limit. And
so do you see the same thing with polio? If you pump up the multiplicity, you reach a limit as to how much virus you get at the other end. Is that right?
Polio is one where it doesn't care about the multiplicity. It does not make defective particles. Mm-hmm. So you can pump up the multiplicity, you never get more virus.
Well, it doesn't make any more defective particles than it would otherwise.
Yes, and you don't get any higher titer. So yes, it's exactly the same thing. And maybe it's a similar mechanism. Of course, polio doesn't go in the nucleus.
Yeah, I would. Their mechanism is pretty specific for a DNA virus that establishes these factories in a replication sites in the nucleus. Though I think it's turning out that many DNA viruses that replicate in the nucleus actually do so in a very focal fashion and establish little subnuclear domains that are factories. They may be mechanistically different from one another. So maybe it would all mechanistically be similar in those viruses. But when you talk about RNA viruses that are replicating in the cytoplasm, I would think if their explanation of the mechanism is right, then for RNA viruses, it's going to be something entirely different.
And for pox viruses too.
Yeah.
Right, in the cytoplasm.
Yep.
So it would be really nice to do this with a variety of viruses. You could do this with pox. You could easily put 3 fluorescent protein genes.
Yeah, I'd have to think about this.
Could it be rate, like a limiting factor, and it's just simply limited by the rate of the ability of these genomes or that wouldn't have anything to do with it, right? I guess because you could.
You're thinking
it's not
about
how many genomes. It's sorry, not how many transcripts, but it's certainly just how many unique genomes can be made.
Right. It would just be if you have 10 genomes being replicated at the same time, and versus 3 genomes being replicated at the same time, there shouldn't be a limit of resources in the nucleus that would restrict you from replicating the 10, because after you've replicated the 3 a few times, you're going to have more than 10 anyway.
Right.
Right.
Another interesting question is why this exists at all. Why is there a limit?
Well, they've got a neat suggestion about that right at the end, where they point out certainly in herpes virus you have this recurring theme where a lot of herpes viruses establish chronic infections, and so you might have a situation where there's actually a positive selection for this sort of thing, so that initially the virus goes into a host and it's there at a low multiplicity of infection. There's a very little bit of virus and a whole lot of cells. And under those conditions, there wouldn't be any problem with all of the individual viruses replicating. But then when you get up to a later stage in the infection, there's a higher MOI going in, and it may be advantageous to the virus to limit the number of individuals that can replicate in order to maintain some kind of genome stability, since obviously this is working out.
How come they didn't know this in 1956? This seems to me like there must be a paper somewhere that just did not get good coverage and had nobody referencing it. But could this have been done?
But the trick here is that they can stick in 3 differentially marked viruses.
Exactly.
And keep track of each one individually, or mixtures of them, and
within a plaque,
within an infected cell.
Okay.
All right, and so that technically has not been possible before this kind of stuff. In particular, doing it this relatively easily. I mean, they actually visualize the cells. They can see how many of different kinds of genomes are in the cell by what kind of color it is.
Right. Effectively, they are counting the number of individual genomes of each that arose from each individual virus that went into the cell.
See, I can think of a couple of different ways of having a limited output of virus, okay? I've always thought of it, as I said, as the cell just being exhausted. So that if you stuck in 100 genomes, maybe all 100 of them replicate, but there's a limited amount of there's a limited supply of precursors and that kind of stuff, so you can only get out a certain amount of virus. If you stick in one virion, then only that one virion or that one genome replicates initially, but ultimately you reach the same exhaustion of the supplies. But what this says is that if you stick in 100, really only a limited fraction of those in this particular case replicate. I don't know what the answer would be with pox. There is an very old pox paper. As a matter of fact, Marc, it goes back to 1963, that was 30 or 40 years later actually repeated using slightly more modern technology, that says that the number of viral factories that are set up in a vaccinia-infected cell, a poxvirus infected cell, are proportional to the input, the multiplicity of infection. So vaccinia, poxviruses establish replication factories in the cytoplasm. And these observations were that as you increase the multiplicity, you get more factories. Now, I forget whether that's saturated. If it mechanistically were the same, you'd get that saturating at some level with increasing multiplicity. I don't think it did. So with poxviruses, this may be a different story.
You should try it, Rich.
Actually, the first thing I ought to do is read the literature.
Nah, just do the experiment.
It may already be there.
Alan, you said it couldn't be done with polio, but in fact, you could make 3 different genomes, a yellow, a red, and a blue one.
You could.
And mix them and do this experiment. All right, so you can't have them generated during infection, but you can do the mixed experiment and look at the color of individual cells and see if you get the same thing. So this could be done.
And the only reason they didn't do it that way in this case was because they'd already made this recombination virus, and they could let the cells recombine it for them.
Yeah, so I think the original purpose was to have in the mouse this virus kick off different colors during replication and see where they go in neurons. But to do the experiment in this paper, which is how many genomes can replicate in the cell, you can just make 3 different stocks of 3 different colored viruses.
That's true, yes.
So we could do it. We have a green polio. We just have to make a yellow and a red. I would be very interested to
you
see.
could do that experiment.
But I can't do this math.
It's okay.
You can find somebody to do that.
That's what Lynn said. He said you have to find somebody to help you with the math.
You can do it. Hey, you're in New York. Go down to Wall Street. They're good with math.
You know, by the way, Alan, in the methods, they tell you how they derived this model, and there is calculus.
Okay.
So,
yes, now I see it. Yeah.
One quick question. So these viruses, regardless of the fact that they're only using up to 7 genomes per cell, they're still making hundreds of viral particles per cell, right?
Yeah.
So there's a lot of RNA. There's a lot of translation, but it's a transcription limiting thing, right? That only 7 genomes are getting transcribed, right, into RNA. So could it be that the the promoter has, for example, a low affinity transcriptional activator, and that the quote unquote the randomness of that transcriptional activator binding in the nucleus to the promoter at the front of the virus, viral DNA, sorry, is difficult to do. So that no matter how many you threw in there, could it be rate limiting at its early transcription?
I think you cannot tell because what you're looking at here are colors. So you don't know if the limitation is at DNA or RNA, right? Transcription or DNA replication.
Right.
I don't think you can distinguish that.
Well, the cells have the potential, to make a lot of RNA.
Sure,
right?
no. I think that's a possibility that it's a transcriptional restriction.
It's got to be early at that stage because, otherwise you just have... you still have the tremendous potential to make the protein, the viral capsid protein in the envelope glycoproteins.
Yeah, my view is that it could be, but I could be wrong. If Lynn is listening, let us know.
It's not the number of viruses; it's the strain, right? So the pedigree, I guess. It's got to be early. It has to be whatever is limiting it. It's got to be.
It is early, yeah. It does occur early, and so that makes it harder to figure out how the virus could be doing this. Is it so early? Right? I don't know how that would work, but their idea that maybe has to do with these nuclear domain 10 bodies may be right.
Actually, the dynamics of the Cre recombination in the infected cell are a sort of a separate issue. Now, this is a little kind of punk, okay, because it's not something that ordinarily goes on. But it's interesting that the Cre recombination happens only before the viral DNA starts to replicate.
Yeah.
And once it starts to replicate, it seems inaccessible to the Cre recombinase, or at least that was the impression that I got from the paper.
That's right.
So that says something about, I guess, maybe the structure or localization or availability of the genome before and during replication.
Could that be what's limiting? And that this is the number of 7 comes from that mechanism of generating the different colors, and that's not really a natural state of the virus, that they got number 7 because the recombination required to produce these different colors is the rate limiting step?
No, because when the viruses are produced from the first infected cell, it produces 1,000 viruses of different colors.
Okay.
Those
that number 7 comes out of an experiment where they took viruses that were already the 3 different colors and did the infection. So that also was independent of the Cre recombination.
Yes,
and if you had just clogged up the Cre recombinase mechanism, then by default, you just get extra red viruses.
All right.
Yes, that's a good point, though, right? We mix the viruses to do this experiment, so it can't be at the level of Cre. Yeah.
Right. I tell you got to read this paper about 6 times.
Yeah.
Yeah, it's a hard paper, but it's very good. It's good.
You would think it wouldn't be that hard, you do 3 colors, just some cells, whatever.
No, there's a lot of subtle stuff, and it's really a lot of basic virology here. So our listeners may find it hard, but here's the take-home message: no matter how many pseudorabies viruses you put in a cell, you only get 6 to 7 of those genomes replicating, and we don't know why, and we don't know if all viruses use the same mechanism. And that's really interesting.
Yeah.
So that's why it's a cool paper. You like that, Rich?
Yep.
Good. I thought you would.
Not just plaques, but colored plaques.
Colored plaques. Nothing better for Rich. Plaques are great.
Plaques.
But when you color them, wow.
Absolutely.
Even better.
Brainbow. All right. There you go.
You should see the math in the method section.
Yeah.
Yeah, that's what I was. Well, in fact, as I was scrolling down to that, I also noticed that 3 of the authors on the paper are in the math department.
Now, let's point out that when you're at a place like Princeton, that's what you can do.
Yep.
You can call your buddies up, and if they're good colleagues, they'll say, yeah, we'll help you. Of course, they may say, I'm too busy, I can't help you, and then you never get the paper published.
The world's getting smaller and smaller, If you're enterprising, you probably do that kind of stuff from anywhere,
Well, I think you got to be convincing, too. Like Lynn is a convincing guy. He can walk into the office and say, oh, we have this cool result. We need your help to look at it. Look at this.
Well, you show up these colored plaques, I'll do anything.
You just got to get the right person, But you can imagine it goes either way. But I do think, I agree, you should collaborate. It's the way to move stuff forward.
Well, it's a fundamental question, right? How many genomes can be replicated?
Yeah.
I'm surprised, again, that this kind of data wasn't generated, I guess, technologically limited, 1956, 1960, basically. But this is so fundamental.
Yeah, it's a basic question. Okay, now the next story is we're getting back to the Australians, who we've discussed releasing various modified organisms or releasing various organisms into Australia that weren't there before, like rabbits and then myxoma viruses to get rid of them and then other viruses.
And because these releases keep going so well, they keep doing more of them.
Yeah, I'm amazed. They keep doing it. And, of course, we talked about the killer pox virus that they made in Australia a couple TWiVs ago. So the Australians are quite brave, I would say, in this sense. So what they've just done is to release a modified mosquito to try and curb the spread of dengue. And I think a couple of listeners sent this story along. There's one in the Australian. Mosquitoes bearing bacteria will be let loose around cairns today as part of a trial to rid the area of the insects that carry dengue fever. So I sent this to Alan, and he found the journal article on which this is based. And this is very interesting. It's based on putting Wolbachia, which are bacteria. And we've talked about Wolbachia before on TWiV. It turns out these are bacteria that reproduce in a number of insects. And
a large number of insects.
Huge. And I think other things too, but for this purpose, insects. And they have some kind of symbiotic relationship such that their presence is selected for, right? So once they get in, they stay there. I think they help reproduction. So they're called reproductive parasites. They can also influence fitness. So a lot of species have these Wolbachia. And I think in TWiV 61, we talked about how putting Wolbachia into Drosophila makes them more resistant to virus infection.
Right.
Now, not too long ago, a group or several groups perhaps found that putting Wolbachia into various mosquitoes decreased their lifespan. So this is exciting because people thought, well, make mosquitoes live shorter, maybe they won't be able to transmit viruses effectively.
Might just make them angry.
Well, it's a little funny because for most insects, it's a positive influence. But I guess if the Wolbachia are not naturally present in the mosquitoes, there's a reason for that.
This may be why the mosquitoes don't normally carry it.
Exactly. So now two separate groups, one in Australia and one at Michigan, have both shown that if you put Wolbachia in the mosquito Aedes aegypti, you can seriously impair the replication of not only dengue, but chikungunya virus and also Plasmodium, malaria parasite.
Now, there's an important difference between, I guess it's a strain difference between these two papers. And I had actually not seen the Cell paper from 2009 from the Australian group. The one that I pulled up was the PLOS pathogens article from
Michigan, right? Yeah.
Right, from just April 2010 from Michigan. And interestingly, the one from the Australian group, they found they've got this strain of Wolbachia that shortens the lifespan of Aedes aegypti and also provides this immunity, pan-viral immunity, apparently. But then the Michigan group is using, they do a similar experiment, and they find that they get the immunity, the resistance to dengue at least, but it actually lengthens the life of the mosquito.
Right.
And
10% of the females, right? It's just like a subpopulation, but it's crazy. I don't know. Maybe it's a timing thing.
Only 10% of the mosquitoes have an extended life. Is that right?
But it's a long tail.
Yeah.
Yeah, this graph in, Figure 6 has this
Right.
long tail on the survival curve. You get down to a plateau where there's only about 10% of the mosquitoes and then have an extended lifespan. I can't explain that.
No. So it's good for some of them.
I think they discussed that in the discussion, and they attribute it to the strain in part, right?
Right.
The cool thing, one of the things we should point out is that the Wolbachia are only transmitted via the eggs.
Right.
From the female. So if you put, the idea is that you put mosquitoes, you release mosquitoes with Wolbachia in them, and maybe they will limit transmission of viral diseases, in particular dengue or maybe even chikungunya and malaria. There's some very convincing experiments in these papers. They basically feed or inoculate mosquitoes with dengue or chikungunya, and they use mosquitoes with or without Wolbachia. There are quite a few controls here. And it's pretty striking. The numbers are almost zero for dengue, for example, in the cell paper, with Wolbachia. Almost zero, except at a very high inoculum of virus. And then without Wolbachia, they actually treat the mosquitoes with tetracycline to get rid of the Wolbachia. And they're quite susceptible to replication with dengue, with chikungunya, and also with a malaria or a plasmodium. They use Plasmodium gallinaceum. It's not a human pathogen, but it happens to grow in Aedes aegypti, and that's where they have their Wolbachia. So they decided to look at that.
But
the methods in this blow my mind, because they're looking at infection of different mosquito parts. So they got people that are dissecting mosquitoes. I can't imagine spending my time doing that. And then the transmission study has to do with infecting a mosquito and then having that mosquito stick its proboscis in a sugar solution and then titering the sugar solution.
Right.
They somehow pump the mosquitoes up so that they're going to salivate more, I guess by not feeding them for a while. So that when they can get them to stick their proboscis into a sugar solution, they'll actually salivate into that solution, and then they can titer the solution and see how much is in there. And they make they draw conclusions about the ability of the mosquitoes not only to replicate the virus, but to transmit it based on experiments like that. My God, really tedious stuff.
Yes.
We had a guest last week on FIB, Brian Kennedy. He was the president of the Buck Institute in California. He was the fellow who discovered the sirtuins in the Garanti lab at MIT. He dissected Cerevisiae. This is a rat hole, it's a classic rat hole. But he took Cerevisiae. In Cerevisiae, they bud and form daughter cells.
Mm-hmm. Right.
And he found that if you tease off the daughter cell, you could get that yeast cell to do it about 20 times, produce a new one, and then tease it off, new one, tease it off. So he's taking a yeast cell, right, and teasing off the daughter cells to count to get a lifespan of the yeast, the individual yeast.
That's kind of standard for yeast biologists with tetrad dissection.
Well, that's tetrad dissection. Those are much larger. He's going in with the single cell.
That's true.
Teasing it off, dissecting, and then he did it 6 million times.
Right.
He did.
That's the tedium. But he found a whole. Well, anyway, you can listen to that episode. But this mosquito stuff is brilliant, though. It is to be able to do all these manipulations on the organism itself and look at all the physiological and pathological studies.
They're very convincing. I mean, there are big differences between with and without Wolbachia in terms of viral replication, spread in the mosquito, transmission.
Yeah.
So we should probably point out that Aedes aegypti is not one of the natural hosts for Wolbachia.
Right.
So they had to sort of tweak this whole thing to get it into that mosquito. And they also found, one of the papers found that if they infected albopictus, which is another dengue host, with at least one of these strains of Wolbachia, they did not get the same effect.
Right.
Right? So
the albopictus is not a great host for
It's
dengue.
not a great host.
Yeah, I understand.
Aegypti is the major one.
So that may be okay.
So then the question is why, right? And
yes.
They did a bunch of experiments that basically come up with a suggestion that the Wolbachia is stimulating the immunity in the mosquito in a fashion that winds up fighting off the dengue infection.
Right, like a generalized antiviral response.
Mm-hmm.
Yeah, in the Cell paper they say, we have this sort of a increase in. They look at a fixed number of innate modulators, and they say, yeah, sometimes it's higher, sometimes it's not. There's huge variability, so we can't prove that this is the mechanism, but it may be. That's what they say essentially. In the other paper, really it's the same. They have they look at half a dozen modulators, including defensins and some other antimicrobial peptides, and those go up, but many of the other genes barely go up. So I'm not sure. The defensins or antibacterial peptides. I don't know what effect they would have on a virus infection.
Let me ask you a quick question. See if I'm getting this straight. So by having a mosquito that's carrying this Wolbachia symbiont, the ability of dengue to infect the mosquito is reduced, and hopefully they're hoping to use Wolbachia infected mosquitoes as a population replacement to eliminate or to reduce the numbers of mosquitoes that are actually carrying dengue and other viruses.
Yeah.
Right.
Yes.
That's basically it.
Not only that, but there's something about the, I'm going to have trouble reproducing this, the biology of the Wolbachia that makes it so that it's preferentially transmitted. If an uninfected male tries to fertilize a female, the egg from a female that's infected, it doesn't work, or the other way around.
Yeah, there's
like
male
that.
killing.
Right. So it's preferentially transmitted.
So it's called cytoplasmic incompatibility, the inability of Wolbachia-infected males to successfully reproduce with uninfected females or females infected with another strain.
Right.
Yeah, so I don't know, since this Aedes is not naturally infected with Wolbachia, I don't know if all this...
We don't know if all that'll all pan.
Yeah,
right.
So, but the funny thing is that just this week on the 4th of January, they released these mosquitoes in Australia.
Go for it.
They put 10
Right?
per household. They identified some houses, and they released 10 per house. 600 different houses.
They said people were, yeah, all right, sure.
I'll do it. Hey, so Rich, if you had dengue there in Florida, and they asked you if they could put 10 Wolbachia-infected mosquitoes in your house, what would you do?
In my house, I'd say sure. But that's my house, right?
Yeah, but if you had young kids, I bet you might think twice.
Yeah, actually, yeah.
I just don't know. I mean, I know so little about this. They say in this article that the CSIRO found the risk to people was negligible. I'd like to know how they determined that.
And I want to know what the follow-up's going to be. All we really know is that they released them, right? So what's the experiment?
Well, the experiment would have to be a subsequent capture where you see how much of the population is carrying Wolbachia. And then also, obviously, you'd be tracking dengue infections, but mainly I think you'd want to see that the Wolbachia survived in the population. And that's where it becomes pretty significant what strain they used, because if they're using the one that shortens the lifespans of these mosquitoes, then they're kind of working their way up the wrong side of the curve there.
Mm-hmm.
It's not ready for translation. I'd say what the most interesting thing is that there's an observation that having this Wolbachia improves the immunity of the mosquito. Finding out what is triggering that immunity is far more critical, because then you could just potentially stimulate it with a small molecule and just, make these. Well, then you'd have these virus resistant. You wouldn't have a natural, population control of mosquitoes other than bats. But I don't know. But it seems early. It seems like this isn't solved because they were able to find a really interesting finding that you could lower infectivity of dengue virus in the mosquito. That's wild.
Right. And I think maybe some of the rationale was that the potential problems, at least to a first approximation, it doesn't seem like it could be that bad. I mean, I always hate to say what could go wrong, but
Yeah.
but really, what could go wrong? I mean, you've got these Wolbachia is in many species of insects. It's floating around out there already. The bacteria are out there. The mosquitoes are obviously out there. So now you're taking the Wolbachia, you're putting it in the mosquitoes, and you're releasing the mosquitoes. It seems like the worst case is that it doesn't work, that these mosquitoes don't compete well in the environment and they die out.
Yeah, I suspect that's what's going to happen. I bet they just die out.
Yeah, that's my money's on them just dying out.
That's what you have when you have a premature translation of a discovery into an environment where you're trying to either a clinical environment or here a sort of a pest control environment. You're going to get a false negative. Well, whereas the 10 mosquitoes that you released create a small little island of resistance to dengue, but when you've got 100 million floating around in your backyard because of a puddle,
Right.
So, Mark, to a biotech person, this is not something you would put your money on right now.
No, but I would certainly, if I was Gates, put money on these guys' research,
Which
not
they
necessarily
did,
on
right?
the translation.
Yeah.
Yeah, absolutely.
I guess it's a good way to spend his money, right?
The more you know about the immunology of a mosquito, I mean, fantastic. And this is a huge leap forward, I think, if you can find anything that can trigger immunity. But dengue virus, here's my question as well. Another question. Why is this virus infecting both the gut of a mosquito and a human? I mean, is the receptor that general? I mean,
Well, that's a good question. As far as dengue receptors go, many have been identified, but there's no consensus, and there may just be many receptors. So it could have a very broad host range, and that's why it does, as you say, the mosquito and humans.
So this is common. This is a very newbie naive question.
No, there are a number of viruses that are vectored in this way, and they replicate in the mosquito as well as in humans. Yeah, that's not uncommon.
Okay, so you could have specificity just for the vector and the human or the specific animal or mammal. You can have, or could this, if you took this, would it infect the gut of a slug?
No, these are pretty specific host pathogen relationships. Okay, where there's a particular mosquito and a particular vertebrate host. Okay, that maintain this cycle through the insect vector and the vertebrate host.
Right, and it's even there's some species specificity, as you alluded to earlier. Not all strains of mosquito, not all species of mosquito are equally good at carrying dengue, and in fact, some of them can't.
Think of the proteins involved in mediating the infection. That's just brilliant to have two completely unrelated organisms.
Yeah, you make a good point. Insects and vertebrates are really quite different, and so for a virus to adapt to replicating in both of those hosts is a good trick.
That is neat. That's really cool.
Yeah, viruses are pretty cool in that way.
But clever. If you want to get around, what better way than a mosquito?
It is nuts. It's just great. Scary too, a little bit.
So Peter Walker, a listener, sent an article that the release has been done. This was in Sky News, where they confuse bacteria and viruses typically.
Yes.
And here they say mosquitoes infected by a virus to help prevent dengue fever.
Nope.
And then they turn around in the next sentence and say the bacterium acts as a vaccine for mosquitoes.
So let's get it right, guys, please. Yeah. By the way, mozzies or mossies, where does that nickname come from?
Mosquitoes,
I know, but why mozzie or mossy?
I don't know. I mean, it's we're talking Australians here, right? I'm probably going to offend some. Yeah. It's the Aussies. It's the Barbie. Is the barbecue. I don't know any others. Okay. They're into E stuff, right?
Right.
Mozzie may refer to many things.
It's Australian for mosquito.
And I think when you do Mossie with the S, is it British? Let's
Maybe.
See. Mossie.
Oh, there's a band by that name.
No, there isn't a mosquito there. All right. Anyway, those are our two stories. Let's do a few email The first is from Viniaka. Some additional information that I gathered on viruses on the verge of elimination. It appears that the next virus on the list of the FAO, which stands for, Alan.
Food and Agriculture Organization.
Thank you. To eradicate...
they're a branch of the World Health Organization, or they're a branch of the UN.
They're heavily involved in the eradication of rinderpest, right?
Yes.
Next virus to eradicate is PPR virus, Peste des Petits Ruminants. There you go, Mark. A little French for you.
Ruminant. Yeah.
In fact, I heard from an Indian virologist friend that the government of India will begin its campaign this year. It is a burning problem in South Asia, the Middle East, and Equatorial Africa. So this is a virus related to rinderpest, which in turn is related to measles, which causes a highly contagious disease of goats and sheep, characterized by fever, necrotic inflammation of the mouth, enteritis, and high mortality. So apparently, this is a economic issue because goats and sheep are eaten, and therefore it is targeted for eradication. I hadn't known that, but thank you.
It's a nice little publication from the FAO that describes it quite clearly. And they also describe the new vaccine that they've developed for this, that they've stuck green fluorescent protein in so that they can differentiate between animals that have been naturally infected and animals that have been vaccinated, which is an important...
That's cool. Something you can do in animals and not people. Next one is from Laura. I was listening to TWiV 104 today, which discussed recent research on colony collapse disorder, a topic of obvious immense significance to everyone. You mentioned the work of Dr. May Berenbaum et al. On increased ribosomal RNA fragmentation among bees of affected hives. Then you went on to mention the lack of consistent correlation with the picorna-like viruses those researchers gave as a likely explanation for the finding. You followed with the pathogens du jour, IIV and Nosemia, which don't account for the ribosomal RNA evidence. That was one of our top 10 stories of 2010.
Right. Yep.
The dual infection. You expressed your gut feelings that something possibly environmental as yet undiscovered may be the unifying explanation for the bee's susceptibility to so many pathogens, but you remained puzzled why an environmental factor would create an illness that arose abruptly and spread like an epidemic. As ribosomes are intimately related to prions, it is both puzzling and disturbing that there has been no visible interest, going by published articles, in seriously examining a possible prion cause. Researchers at the University of Malmö, Sweden, have made great strides in amplifying prions and identifying them from tiny samples. Their technique could be applied. The finding of ribosomal RNA fragmentation could be the smoking gun of an apian prion disease. Ignoring prions is such a glaring omission that it makes me wonder if this direction of inquiry is for some reason being suppressed. Possibilities include the lack of known treatments for such diseases, the huge expenses associated with the potential need to destroy hives all at one time in large areas to contain such illness and start afresh with colonies from the few unaffected areas like Montana and Australia after burning and replacing all the current housing and perhaps most equipment, and the public's possible rejection of honey at the very time beekeepers would most need income. People often avoid explanations that demand very difficult solutions. However, scientists are a group who include more than the average percentage of pragmatic individuals. You understand that as insecure as a difficult solution may be, the only thing worse is no understanding and no solution at all. Could it be that research money is only available to find palliative answers to allow colonies to hang on for the pollinating season with yearly replacement without eradicating the underlying cause? If it turns out to be the case that the only promising explanation that has not been eliminated is the kiss of death to a researcher's funding, would there be any point to investigating further without speaking out? I would very much like to hear your thinking.
I think we don't need to go quite that far in theorizing why people haven't looked at a prion cause for this. People are looking at all sorts of causes for colony collapse, and they're finding all sorts of potential causes for it. And I think what we've touched on a few times is that this is probably multifactorial, or it's a case where the bees that are being trucked around the country for pollination simply have lower resistance to a lot of these pathogens. I don't know. It seems like you'd have to go a long way to see some kind of conspiracy here.
I don't know why you have to. So science works. Science is largely investigator driven, right?
Right.
Somebody thinks of a problem they want to solve, they go at it. I don't know that there's any suppression. Now, I know people are going to tell me I'm wrong, but when you have a lot of scientists who have big egos and are very aggressive working, eventually they get to the solution.
And if I were to try to select a group of people that would be least qualified to pull off a conspiracy, it would have to be scientists. I mean, I'm sorry. You can't get these people to conspire on a friggin' pizza order. It's.
I agree. I'm okay with this letter up. I mean, I think the entertaining the notion of a prion being involved in this is an interesting idea, and I'm okay. I'm okay with a letter up to the point where we're talking about the notion of the inquiry being suppressed, because I just don't see that kind of stuff happening. It's just.
So let's talk about the prion aspect. That's why I put this in here, Mark, because I know you love the prion.
We've covered it a couple of times. The listeners can go to Futures Biotech episode 1 and number 57, and I'd focus more on 57 because we've had Susan Lindquist. Now, Susan Lindquist is a chaperone person who studies prions as well. Prions is an unfolded state of a protein. And what she finds is that there's an interesting correlation between sort of the environmental stress and the ability of chaperone, one in particular, which is HSP90, to sort of buffer the environmental stress by stabilizing proteins in the organism. But then if there's a threshold of environmental stress that is reached, that chaperone no longer has an ability to prevent misfolding. And what happens is the HSP90 specifically targets transcriptional activators. Those are the dip switch proteins that go and bind to the genome and turn genes on and off, right? So the letter is, somewhat plausible in that an environmental stress could lead to the chaperone's inability to handle the environmental stress, triggering a spew of morphological change because the transcriptional activators are now binding to the genome in many, many different ways, allowing different phenotypes to occur, which may be less resistant overall as a population to the environmental stress, right? And making them more susceptible. That being said, the link between, for example, you take HSP90 out, protein misfolding happens. You have prions, which are in an unfolded state of the protein, forming and sequestering out specific transcriptional activators. For example, you can sequester out the suppression. What's it called? SUP35. You can. There's a protein that stops transcription. Right? It says this is the end of the gene, stop here. And that is actually a prion. And if you remove it and sequester it from a folded normal protein into a prion fiber, now when you're making that gene, you're making a whole bunch of different versions of it. And it never knows when to stop, and you get all these changes, right? So you can have prions related to environmental stress, which leads to significant organismal change. You can have heat shock proteins with a threshold that can be attained, that if you go beyond that threshold through environmental stress, you can now have organismal change. So those fundamental concepts are plausible as to the susceptibility of the bees to pathogens. But then the next thing is, well, sure, it could be anything.
Yeah.
But there's in all of this, is there actually a connection between prions and ribosomal RNA degradation? I don't see that.
So what I looked a bit into this, and it turns out that the ribosome has protein folding activity. All right. Mm-hmm. Apparently, it can participate in being a chaperone, and there are some drugs that inhibit prion formation, which happen to target this protein folding activity of the ribosome.
Okay.
So there's some connection between the two. To inhibit prion formation, you can just inhibit the protein folding activity of ribosomal RNA. Now, what we see in colony collapse is a degradation of ribosomal RNA. So that would take away the protein folding activity. So that would inhibit the prion, because the drug takes away protein folding activity. That inhibits prion propagation. So I don't know how degrading the ribosomal RNA would help propagate the prion in these bees.
Right. And another question, has anybody found prions in insects?
That's a good question. Do you know, Mark?
I'm sort of having a one of those flashback moments right through that.
Yeah, Drosophila.
There's prion-like proteins. There's proteins related to those in yeast that have been done that are also in insects, but have they been demonstrated to be prions? I mean, it's such an early area of research. There's two main researchers that I think are producing the most interesting prion work are Lindquist and Kendall. Prions are Kendall's, when he presented in Neuroscience last year, he showed that there's this one protein that by sequestering it resulted in sequestering the protein into a prion state led to long-term memory potentiation. So prions might not necessarily be pathogenic. There's examples, for the most part, there's examples where they're not a pathogenic state that leads to neurodegeneration, but can be part of our biology in mediating the long-term memory potentiation. Lindquist's sort of scan of the genome, the yeast genome, for all the potential prion-like proteins, and then verifying that several are, show that prions are far more prevalent than we expected, and are even potentially part of a non-Mendelian form of inheritance, right? Forming a significant contribution to evolution. But this is really, really early stuff.
Yeah, you're right. There are prion-like proteins in insects. Whether they have the properties as the yeast prions do, we don't know. But
whether they can be pathogenic.
Yes,
that was really my question.
Yeah, and that's a good question. And I think
we should probably point out that prions are inherently a huge pain in the tuchus to work on.
They are. They're difficult.
Well, that's one of the things that she's pointing out here in this conspiracy theory, is that if it were prions, they'd be difficult to deal with. Okay, because they're hard to inactivate, and et cetera.
Well, they're hard to attack from a combating disease standpoint. But what I mean is they're difficult to study.
Yes.
So if you're looking for a reason why there isn't a whole lot of work on apian prion disease, it may just be because it would take you decades to set up.
Right.
Yeah, they're really stable though. I've worked with amyloids and amyloid fibrils, and did a lot of microscopy, cryo-EM and stuff with them. And the great thing is you could make a sample of them, right? And they'd last on your bench for a year, and you'd have enough, EM doesn't require a lot, but it's the most stable form of protein conformation. Once they're made, they're steel. I mean,
that's part of the problem, right?
Right. Yeah. You're twisting the backbone, the protein backbone, away from a natural protein conformation to being a sheet, right? With the what do you call the peptide backbone forming the bonds that form this incredibly stable conformation that any protein can form. So it's extremely robust. So it depends on what you're doing. If you're using them as nails, or as Susan Lindquist did, she started a company she mutated the protein so that there was a cysteine residue in it, and then with a cysteine residue you could label it with a gold particle, right? And now she has nanowires that are extremely strong, and she's selling nanowire like amyloids.
Oh, cool!
Neat.
Yeah, very, very cool.
What's the name of the company?
I don't know. This was an interview I did 4 years ago as episode 1 of Futures of Biotech.
Now, if you wanted to cap off the irony on that, you could build memory chips out of them.
Yeah, you could, but they're not logic gates. They're
I know. It was, yes, they function more like diodes, but yeah.
Cool.
Exactly.
The next email is from Todd, who writes, "Hi, guys. I've been listening to all the previous TWiV and TWIPs since hearing you on Futures in Biotech." There you go, Mark. Currently at TWiV number 90 and have been intrigued with your discussions on junk DNA. While growing up in Canada, the highlight of my Saturday morning was listening to the CBC radio science program Quirks and Quarks. It has had a few wonderful hosts over the decades, giving me a regular science booster until I moved to the U.S. several years ago. I missed it greatly until I found it again as a podcast, giving me a taste home while living here in Denver. This week's 35th anniversary show was recorded in front of a large audience in Toronto, and one of the guests discussed junk DNA, and he included a clip. I'd love to hear your ideas on this and apologize if you have gone over it since episode 90. Vince, Dick, Alan, and Rich, you feel like old friends by now, and though you don't hear me, I'm constantly poking my comments into your conversations. Keep TWiV and TWIP going. You are an incredible source of information and entertainment. Take care and happy holidays, Todd. So we have this clip from Quirks and Quarks, November 13th. Do you know this show, Mark?
Absolutely. And Bob McDonald, the host, is fantastic. He really does a lot of preparation, and he brings in a lot of interesting topics in the area of science. He's just outstanding. He's a really great communicator.
We should point out that Mark, of course, is from Canada.
Yeah, I used to watch Bob McDonald. Remember, he did kid shows. He did everything. He's really a great science communicator in Canada.
It's not Captain Kangaroo, is it?
No. Was that in the U.S. too?
What, Captain Kangaroo?
Oh, yeah.
Yeah, it was.
Cool.
Forgot the guy's name. I thought it was Mick something.
Mr. Dressup?
So I played this, and so it's very vague because she doesn't really specify anything, but she talks about how we thought there was junk DNA in the genome, but now it's important for gene regulation. And I think she's talking about small RNAs, basically, like microRNAs, which are known to regulate gene expression. But she never identifies them as microRNAs. But I think that's part of what the issue is, that a lot of what we thought was junk is now regulatory stuff. But she didn't touch on the endogenous retroviruses, which is a good fraction of this so-called junk DNA and what they do. And we've talked about that a little bit. We don't know if they have function. They've been there for years, and some of them might have functions.
If I had to guess, I would guess that there is a sizable fraction of DNA that is genuinely junk, but there's also probably a lot of DNA we don't know what it does now, and we're going to find out down the road.
Yeah, I would agree with that, because you also have examples of organisms that have more or less of this stuff, which suggests that there is a certain amount of wiggle room in it.
Right. Fugu fish, right?
Yes.
Right.
Well, if you think about Jupiter, right? It's what a disconnect. It's moving around in our solar system, teasing out comets and preventing or cleaning up our solar system by basically creating a vacuum for comets. Junk DNA, I guess, is great if you can have viral infection, retroviral infection. You don't want it to go and disrupt something important, right?
Yeah. It's a cool analogy.
Interesting idea.
I like that.
Who knows? Yeah,
that's why we have Mark here. Next one from David. I discovered TWiV this summer and I'm doing my best to catch up. I have a short 15-minute walk commute, so it takes time to get through all these episodes, but I look forward every day. With Rich Condit on the show, this comment is probably unnecessary. He talks about the passing of Frank Fenner. Although I never met him, I consider him one of my science heroes, someone whose work has greatly influenced my career and interest in virology. It would be great to hear some discussion about his contributions, which of course we did. It has also started me thinking about the people who influence us in our careers and stimulate our interest in science. Of course, our teachers and advisors are among them, but then there are the other scientists whose work or life stories influence our decision to go into science. Certainly he was one for me. Another was Linus Pauling, who died when I was in high school, and my chemistry teacher wore black for several days after his death. I wondered who this guy was, so I read his biography, and it remains one of the most influential books I have read. Luckily, another one of my heroes, David Suzuki, is still alive. Your Canadian listeners will know him well. I'm curious about who the most influential people have been in your careers, especially in terms of stimulating your interest in science or virology, or whose work has had a major impact on your careers. And David is a professor at Vassar College. There's a link to his website here, and apparently works on pox viruses. I don't know if you know him, Rich.
Yeah.
Who? Let's go around just quickly. Who was a hero for you guys, and like David is suggesting?
Well, I thought about this. Actually, most of my heroes really are, or the people who really influenced me, are the people he kind of excludes from this discussion in a way.
Your advisors, right?
The advisors and stuff. You know, I feel very fortunate to have had a string of teachers and advisors who were really great role models. They were terrific and had a very positive influence on me. If I think of somebody who, you know, was not really a teacher, who was an inspiration, though I would, the first one that comes to mind is Fred Sanger.
3 Nobel Prizes, 2 papers.
Two.
No, he won 2 Nobel Prizes. Sorry, that's right. But he had 3 scientific papers in 1 decade. And people wondered why he didn't win the 3rd Nobel Prize with that 3rd paper. Right? What was wrong with it?
Yeah, he figured out how to sequence proteins, and sequenced insulin, and they gave him the Nobel Prize for that. Then he figured out how to sequence RNA, and they didn't give him the Nobel Prize for that. And then he figured out how to sequence DNA, and they gave him the Nobel Prize for that. And a very unassuming individual, low key, as Marc has pointed out, you know, didn't run a big factory, or says most of the work was done with his own hands, with you know, a couple of collaborators in his laboratory. And I just really admire his style and his intellect. It's just incredible. So yeah, he was a role model for me.
How about you, Alan?
I'm kind of in the same boat as Rich, where my really big inspirations in science were teachers I had along the way, and to some extent family. My grandfather, who died before I was born, but I was familiar with his work, and he'd been a researcher. And then, you know, other than that, you know, I was always inspired to a great degree by the teachers along the way, and I guess the stories of the greats. You know, Charles Darwin, Louis Pasteur, certainly resonated, and I thought, wow, you know, that's really, really cool to have lived a life like that. And what's the modern equivalent, and how do I sign up?
Marc.
Yeah, I've got a few, but I'm going to go the other direction. So, well, David Suzuki, who had a show called The Nature of Things in Canada, inspired a generation of scientists. Most scientists that are between the ages of, I don't know, 20 and 50 in Canada right now got inspired in part by him raising questions. That show wouldn't so much provide answers, but raise questions and get you really interested in those questions. And you'd go off and, you know, try and inquire and learn everything about the topics that they covered. So he was brilliant, but not a scientist per se. He's a geneticist, but not, and he wrote a textbook, but he's not the scientist that inspired me. I've got a couple. One, Mario Capecchi, who I had recently on Futures in Biotech, was a homeless child in Italy during World War II for 5 to 6 years, hadn't taken a bath in 6 years, and then went on to win the Nobel Prize. Right, he did his PhD with.
There was some stuff that happened in between those two.
Yeah, but what an incredible story! I mean, going from homeless child to, you know, no formal education as a child, right? No elementary school, nothing, to really being able to answer some fundamental questions and provide a technology that's changed medicine. That is to create the knockout mouse. But that's not my number one scientific hero. So the next runner-up is Rick Lifton, who's the chair of genetics at Yale, whose discoveries on hypertension changed how diuretics are used, allowed diuretics to be used for hypertension. He basically discovered that hypertension was not a disease of the heart, but a disease of the kidney, an ability to handle salt, which resulted in hundreds of millions of people having an improved quality of life and survival, right? So the ability to affect the lives of hundreds of millions through scientific discovery, he's a great example. So that's Rick Lifton, a chair at Yale. But the one who really inspires me is I had him on Futures in Biotech episode 23, and that's Buzz Aldrin. And now Buzz in the media is portrayed as kind of a guy who's going after, the camera, right? And really seeking out attention. But, as an engineer, so here he went to Korea, flew 66 missions, went after Korea, went to MIT to do a PhD. And we're talking in the late 50s. In the late 50s, he wrote a PhD thesis at MIT on how to dock spaceships. Right?
Wow.
Then in the 50s, then in the early 60s, he's flying spaceships, docking spaceships in the Gemini program. Just absolutely insane. And then, his what he had researched and developed turned into be one of the fundamental techniques used in the Apollo missions and getting us to the moon. And then he flew to the moon. So he went in the 50s from, imagine this, right now, it's kind of like writing a thesis on teleportation today, and then in like 2 years from now, teleporting, right? He's so forward thinking, and a brilliant engineer. That the idea is that you can do anything, and this is in the 50s. He's flying spaceships basically in his head, and then within 10 years made it happen. My work is translational. Right, I'm trying to get what we understand from the lab into the clinic, and it's a great challenge. It's a huge challenge. But if there wasn't guys like Buzz Aldrin who did these giant leaps of faith and then got it to happen, then why would I even bother trying? Right. So he kind of inspires me that way.
It's cool that you could do a podcast with him, right?
I was running around Yale going, I just spoke to him. I can't believe it. Another guy, Harrison Schmitt, he was Apollo 17. Brilliant guy. More understated, but the only scientist to actually fly to the moon. If you want a first-hand account of what it feels like to stand in the valley of Torres Litro and look up, he was on the moon for 22 hours EVA, right, outside his vehicle. So nobody has lived on another celestial body more than Harrison Schmitt. So you can listen to that episode too. But he tells you that you're looking up at the sky; it's pitch black. This is not kind of written in the books, and so we got it on tape. And he says the only way to see a star when you're standing on the moon is to make a circle with your hand and create a 1x telescope to reduce the dynamic range of light, or to expand the dynamic range of light that you can see in your eye. But anyhow, wow. Buzz is the guy. He's my hero.
Wow.
He did it. He did the impossible. So.
Well, I do plaque assays. I had lots of great advisors, and Peter Pelletier, David Baltimore. I had a great high school teacher, but maybe 2 books I read really made the difference. Microbe Hunters by Paul de Kreef, and Fever, about Lassa fever outbreak. Those are 2 that really inspired me to go into science, and in particular virology. There you go, David. And last email is from Kimberly. I was listening to Futures in Biotech episode 71, where you described the recent PNAS paper on TRIM21. You had mentioned that this antibody TRIM21-mediated virus destruction may be why viruses such as polio and flu only need a few antibodies for neutralization. This could work for polio, but probably not for flu, as it is an enveloped virus. The paper states many times that this can only work for an unenveloped virus, and it took me a bit to realize why, as they didn't spell it out. If the antibody is directed to the viral glycoprotein or any other protein on the outside of the virion and binds to the virion outside of the cell, as the paper describes, then upon membrane fusion the antibody would remain outside of the cell and inaccessible to TRIM21. I think only time will tell if there is a similar mechanism for enveloped viruses. Thanks for the podcast. Kim is a postdoc at Johns Hopkins Bloomberg School of Public Health. So I will direct everyone to Futures in Biotech episode 71 to understand what Kim is talking about. But it's basically a protein, TRIM21, which we think is involved in antibody neutralization of viruses. And she's got it right on here as to why
sounds like it
might not work for envelope viruses. So thanks, Kim.
By the way, quick note to Kimberly, if she can get me in touch with Peter Agre. He's there.
You want to do a podcast with him?
Yeah, he's great. No, I actually met him last summer, and he's going to be. I haven't contacted him yet, but he's already agreed to be on FIB.
All right.
So that'll be a fun one for her. Yeah.
All right, let's do some picks of the week. And Marc, let's start with you.
All right. So I've got to get out a catalog here. So I thought I'd go hardcore science on this. I went around the lab and thought, what piece of equipment do I really enjoy using and what I think is really cool in the lab? And it's the anti-scientific piece of equipment. One of the things we need to do in biochemistry, right, is to make a lot of purified protein. And you need milligrams, if not grams, if you're going to do stuff like crystallography and try and determine the atomic resolution structure. So one of the things, though, is I have a small company, and it's very hard to pay for the $100,000 bioreactor or fermenter, or the $50,000 cheap unit. Even that's a struggle because I have to get a loan and put up my house as a collateral. So what I did is I flipped through a Corning catalog, and I found this big jar. It's called the ProCulture Glass Spinner Flask with angled sidearms. Well, not the angled. Use the straight sidearms. Okay, so these are 15 to 36 liter flasks, right? They're kind of like a big jam jar. It's like a big, shaped like a jar of jam, and the lid is a hard plastic. And the thing is, they're $800. And when you want to make protein and you don't have a $25,000 to $100,000 bioreactor, you can use an $800 jar. And in the same catalog on the page right beside it, there's these. There's a set of fittings and a direct drive motor, right? And it's this powerful electric motor that clamps onto the lid of the jar and uses a direct drive paddle. So it has like a triangular paddle that spins inside this giant. It's a jar the size of a barrel for making wine, right? Okay, so I've got a video. I sent you a video.
I put the link in the show notes, guys.
Yeah, I just looked at that.
I'm looking at the picture and I looked at the video. Yeah.
Basically, this setup, if you use the Corning jar and the Corning motor, it's the direct drive motor and the direct drive pallet assembly. You've got the makings of a. That costs about $2,000 to set that up for the start of a bioreactor. Now, the next thing is you need air, right? And the air has to be, one great organism to grow is Pichia pastoris, which is a yeast. And this yeast is particularly interesting is that it doesn't ferment. It is it grows in an aerobic environment, so it doesn't produce ethanol and it doesn't limit the number of cell divisions it can do. So you can get optical densities of up to 30 to 40. So you get this like deep slurry of yeast cells growing, making your protein.
Is that what you have in the picture there?
Yeah, that's what we have.
Oh, man, that's pretty dense.
Oh,
yeah.
That's an OD of 30 there. But you can boost the OD to theoretically up to 400.
My gosh.
It is 50% of the solution is yeast cell. It is obnoxious. So here you've got a $2,000 bioreactor. Now you need to pump air in it. And if you're going to do this, make sure there's a couple of things. You can send me an email at marc@twit.tv, and I'll give you a protocol. But you need to grow the Pichia pastoris to obnoxious density. So you don't want to use dextrose. You want to use glycerol. And also when you're in induction mode, use methanol and use it on every day. Never starve the cells and grow them up for 4 days. Okay, that being said, the air has to be clean. It's got to be breathable air. Most pumps, air pumps, if you're going to do a la cheapo, is not to use an oil pump, which is a typical air compressor that you would get from Home Depot. What you want to use is, so I walked around the lab and I saw a vacuum pump underneath the hood. I just checked it, and of course, if it's a vacuum pump, it's also going to produce air. So at Thermo Fisher, I lost my screen here. Thermo Fisher, they have these pumps called the Maxima Dry. So go to Thermo Fisher, and for about $1,800, don't just buy it. Get your rep to give you a quote, and then say, okay, now sharpen your pencil. But you can get these. They're called Maxima Dry oil-free vacuum pumps. And what you do is you grab the air coming out of the vacuum on the other side, and you throw it into this giant jar. and you put a really good diffuser. The smaller the bubbles, the more the yeast are going to grow. So you put like an aquarium style, diffuser, and
you also have a 0.2 micron filter on the intake.
Yes,
to keep it sterile, right?
Yeah. As a matter of fact, you can buy these little filters, the diffuser that actually act as filters as well. So in Piccia, you're growing it in methanol. So
now, is there a reason an actual aquarium pump wouldn't do this?
You just need something a little bit more powerful. I mean, you could have a series of them and put them in parallel. And I thought about that. Yeah, we thought about some strong aquarium pumps, but I couldn't get my hands on one that would really be strong enough. So I did shell out the $1,800 for the 2-stage 35 liters per minute capacity, and this thing bubbles out. It's crazy. It's absolutely insane. The whole lab smells like beer. Okay, so total cost between $5,000 and $7,000, depending on what you have in the lab. For example, you just take the jar and you put it in a bathtub or in like a plastic tub to keep the water at 30 degrees. If you have a thermal, cycling refrigeration unit, most labs have these around. I'm talking to the young professors out there trying to set up inexpensively. Set that up. $5,000 to $7,000, you can get pretty much everything going, and now you're going to be making grams of soluble protein and high numbers of maybe 50 to 100 milligrams of membrane protein and Piccia pastoris without going broke. And it's working for us. So.
The movie is just water, just to show the impeller, right?
Right.
But
it's nice.
It's aggressive.
I think this is useful. This is a first on TWiV, a real practical pick of the week that you can use in the lab.
Yeah, and I never thought it would work. I was just flipping through the catalog and said, hey, well, what if you could do this? What if you could attach that? And so it's a cheap bioreactor.
Well, this is the thing you can do in a lab. You can build stuff. You don't have to go with what's already there. You can take some risks.
Reminds me of graduate school. We used to grow bacteria in 20-liter carboys in the sink with an immersion heater. With a sparger that was made with a piece of sponge stuck onto the edge of a tubing. It didn't work.
Well, I built a 25-degree precision incubator in graduate school because it's very hard to maintain 25 degrees C without a really, really expensive setup. So I told Vince, hey, I want to just order some like an old computer fan from a surplus store and try this. It's like, yeah, sure, try. And it worked.
Yeah, I remember that. It was good.
I used a magazine to keep the door open. Exactly.
Great.
People are, you get caught up in some of the suppliers and the equipment companies that are selling these $50,000 units, $100,000 FPLCs, for example, where you don't really need it. All you need is a column, a peristaltic pump, and some patience, and you can do FPLC. On your bench. You don't really need fancy gear.
So what should we call this? A homebrew fermenter?
Yeah, that's a good idea.
Or bioreactor.
Yeah. Now don't do this at home. Maybe we should tell people not to do it at home. Don't drink it.
That's nice. I like that. Thanks, Mark.
No problem.
Rich, what do you have?
I want to talk about a Christmas present that I got. Now, my guess is that there are a bunch of tech types out there for whom this will be sort of ho-hum. But for me, it was a shock. So this is a universal remote control made by Logitech. This one, they've got several models. The one I got is a Harmony 700. Like a lot of people, I've got a stack of equipment and a box full of 5 or 6 remotes, right? So it's a cable box, a TV, a DVD player, a tuner, and a CD player. So that's 5 different remotes. And the cable box came with a universal remote. But it doesn't work great. You got to look up the numbers to program all of the different devices, and you can't find them all necessarily. And then you still got to sort of play around with the inputs and all that stuff. This thing, it's like a handheld computer with a bunch of buttons on it. Okay. So you get this thing and you go around and you go to all of your machines that need a remote, and you identify the make and the model number, and you go to a website for Logitech, and you register your product, and it has a bunch of wizards that it has you go through. And the first thing you do is you tell it all the devices you have and their model numbers. And then it goes through and it says, okay, what do you want to do? You want to watch
and
TV?
they show up and rob your house.
No. You say, what do you want to do? You want to watch TV? Yeah. Well, how are you going to do that? Do you want to use component? What input do you want to use? Do you want to use the cable box? Do you want to play it through your tuner? Or otherwise, if you want to play it through your tuner, what input are you going to use? Blah, blah, blah. You answer all those questions, okay, for how you want to watch TV or how you want to watch a movie or how you want to listen to a CD. And then you plug your remote. When you're all done, you plug your remote into the computer and it downloads all this information on the remote. Okay. And then you just take the remote and you push a button that says watch TV, and it turns on all the right stuff and sets it all the inputs up right. And you're watching TV. And if you say, no, I want to watch a movie, it turns off the right stuff, turns on the other right stuff, rearranges all the inputs, and it's got it. And it's brilliant. And if it's not quite good enough, you can go back in and you can tweak individual buttons and reprogram individual buttons. I've never seen anything like that. I threw all my remotes away.
You know who invented this?
No.
Concept? Steve Wozniak. He created the first programmable remote control using the 6502 processor, the same processor as in the Apple 1 and 2. Crazy stuff.
Wow, I didn't know that. Who gave it to you, Rich?
One of my daughters.
Cool.
Actually, the idea, I think, was her husband. And he wanted it for himself. In fact, we wound up getting one for himself, too.
That's great.
Yeah,
Good
it's very
pick.
cool.
Thank you, Rich. Alan.
Well, my pick of the week is something that's actually been online for a couple of years, apparently, but I just recently stumbled across it. It's a complete collection of all of the reports from the HMS Challenger expedition of 1873. And if you're not familiar with this, it was the first global oceanographic research cruise, and I think it was probably the first big science project funded by a government. The major mile point in science history, but it's also there's a lot of really cool stuff here. So people often think of the HMS Beagle as a research cruise, but in fact that had nothing to do with research. It was a mapping cruise, and Darwin managed to finagle his way aboard and managed to do science. The Challenger, 40 years later, the Royal Navy and the Royal Academy got together and funded this expedition, whose main purpose was basic science. They wanted to go out and sample the deepest points in the ocean and dredge up bits of the bottom and see if they could find anything living there, to test one of the fundamental questions of the time, which was, is there a cutoff point in the ocean below which you cannot have life?
Wow.
And so this expedition is sailed around the world, and this was in the days when it's literally they're sailing around the world. They had an auxiliary steam engine, but mostly they were sailing along the trade winds. And they took this Royal Navy ship and stripped most of the guns out. They still kept a couple of them, but they equipped the gun deck as mostly laboratory space. They added more labs on the deck houses. And they went around. They also equipped it with big, long lengths of cable and dredges. And the first, I think it was probably the first water sampling, what's now called a rosette, a device that goes down and samples water at different depths. And they did all this really cool science, not only at sea, but also everywhere they stopped. They would go ashore, and the naturalists would kind of catalog what they found. So this is a site that has all the documentation, and some of it's very inside. I mean, it reports on all the individual classifications, taxonomic classifications that they encountered. But then there are also some general summaries of the cruise that are books, and just absolutely fascinating reading to see how they did this stuff back at a time when the technology we take for granted didn't exist.
Wow, this is some resource.
Yeah.
Great maps. I'm just looking at the front page.
Yeah, I was looking at the maps too.
Oh,
What's
the pictures
the
are awesome. 19thcenturyscience.org. Yeah, and they have plans of the ship and maps where they sampled and neat drawings. And it was the first expedition to use photography as well. So there are actually photos a lot of things.
Nice. Beautiful. I'm surprised we haven't heard that from you before.
Well, I only recently stumbled on it.
Cool. Thank you. Excellent. Well, my pick is two things. First is the sequence of the woodland strawberry genome in Nature Genetics. This is open access, so everyone can see it. And this was actually done by a number of people, including the PI who was at University of Florida, Gainesville.
Right.
I don't know if you know this fella.
I do not.
But this is a 240 megabase genome with 34,000 genes, 34 to 35,000 genes. So it's pretty cool. But what's even cooler is that the PI Kevin Folta has a blog called Strawberry Science Blog. And it's called A Weekly Report on Recent Findings in Strawberry Science. Unusual to have scientists blogging, but here you go. And he talks about the history behind getting this project going, which started.
Is so convoluted.
It's amazing.
Yes.
So he wanted to do it by 454 and looking for $100,000 and had to submit a proposal, but he was competing with peach and apple genome sequencing. It's a really cool story, and you have to read it. So that's my pick of the week.
Right.
Very cool. And that will do it for another TWiV. You can find us on iTunes at the Zune Marketplace. You can also listen on your iPhone or Android device using an app made by microbeworld.org, or you can go to twiv.tv. You can download episodes, play them right there, and check out our show notes. And as always, send us your questions and comments to twiv at twiv.tv. Marc Pelletier is at Futures in Biotech, twit.tv/FIB. Thanks for joining us, Marc.
Well, thanks for the opportunity. It's always fun to talk to you guys.
Always good to have you on. And let's try and do a joint TWiV fib. See if we can make that work.
Let's get that up. And up on video, too. It'd be good.
Yeah, I think Rich and Alan would enjoy the video part.
Sure.
I'm all set.
Alan Dove is at alandove.com. Thanks again, Alan.
Always a pleasure.
And Rich Condit is at the University of University of Florida at Gainesville.
Great to be here. Always a good time.
I'm Vincent Racaniello. I'm at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.